All right, take it away.
All right, thanks, Raymond. All right.
All right, we can see it.
Yep. Hi, everyone. Thanks for being here. My name is Dave, and yeah, I'm from Malaysia, a PhD candidate in Kyoto University, and I'm an epigeneticist. So before we go into epigenetics, I'm talking about epigenetics and aging and how your genes don't really matter. They do matter, but not that much. And before we go into epigenetics, let's talk about genetics. So what is genetics? Genetics is code or template that decides who you are and what you are that you inherit from your parents. And they're all assembled in these packages called chromosomes. So here we have 46 chromosomes or 23 pairs of chromosomes, and the humans have 23 pairs of chromosomes, and they're all condensed into packages. And we distinguish different organisms based on how many chromosomes they have. I'm sorry, different species. And humans, for example, have 23 pairs of chromosomes, and chimps have 24 pairs of chromosomes. And if you stretch out these chromosomes and align them in a straight line from a single cell, how long would that come about? We have humans at 6 feet, and then we have an anime character, Kujo Jotaro, at 6.5. And if you line up DNA from a single cell, it'll come up to about 2 meters, almost as tall as Jotaro. And let's say you take DNA material from all the cells in your body and line them up. The total length would come up to about 6.2 billion kilometers. And that's enough to make a round trip from Earth to the sun 21 times. That's a really, really long material. But imagine that 2 meters of DNA has to be condensed into a compact form to fit into a nucleus of a cell. That's about just 10 micrometer in diameter. How does our cell do that? How did evolution manage to get this far? So I'm sure everyone's familiar with the structure of DNA, the ladder form, but what most people don't know is DNA doesn't really exist in this free state in nature. They're usually coiled around these structures called as histones. And histones sometimes represented like a sphere. In this case, it looks like a disk. So in order to minimize the surface area, DNA is coiled around these structures and then looped into a big chromosome, a single chromosome. And humans have 23 pairs of these. That's how we fit into a cell. This structure was discovered back in 1970s by a scientist couple named Ada and Donald Olens. And they found out that this is a single strand of DNA. And within this DNA in the region, you can see like a bead-like structures. This is actually a real microscopic picture of DNA. And they famously quoted it being beads on a string, not to be confused with Dr. Dre's beads on a string. And this was a magnificent discovery because if chromosome is looped like this around a bead-like structure, not all regions of DNA is accessible for expression. Again, back to a picture of the chromosome. And one of the biggest hurdles we have pre-2000 is we didn't know what genes are or how many genes are there in humans. Because like I said, it's a template of that determines what we are, and we don't know how to read the template or book that has the instruction manual. So they launched the Human Genome Project in the 1990s. The goal was to uncover every gene that codes for the human genome. It took about 13 years from 1990 until 2003. And the results were shocking because scientists thought there would be hundreds and thousands of genes in the human genome. Unfortunately, we only have like 20,000 genes that are actually active, and that's just 1% of the genome. So when you saw the structure of the 23 pairs of chromosomes, only 1% in all of those combined actually codes for something. The rest, 99%, was taunted as a junk region because these are just random combination of ACTG genetic codes. But in recent studies, we are coming to understand that these junk regions are not really junk, and they might have like a bigger role in managing the 1% of the coding genes. So this 99% of the genes are somehow influencing the 1% of these genes. This is a material for another talk, but this is just a basic introduction on how we got to the decoding human genome and why it matters. So, like I said, the human genome exists like a string attached to a bead, and it's tightly strung around this bead-like structure. And I'm sure everyone knows here that genes has to be expressed to give their effect, but they're all hiding in this bead-like structure, so they're not readily accessible because not all genes in our body are being expressed at all times. Some are always off. So some responds to some kind of stimulus. So when there's a stimulus, these bead-like structures will have to physically open up to reveal a region of DNA. So here we have a stimulus, let's just a random heat stress stimulus. What happens is the bead-like structure unravels to reveal the genes hidden within those strings. And you can imagine this like an actual electronic circuit. So here we have an off-state circuit. The bulb is off and the connection is severe. But when you connect the circuit, you actually have like the light bulb glowing. But the circuit is not going to connect itself on its own. You need some kind of stimulus from the outside to push the circuit on. But imagine this. What if there's an obstruction? What if there's a pesky hand blocking you from connecting the circuit? No matter how much you try, you're not going to turn the lights on. Just like that, even if you have unraveled that genetic material from histones, so normally the activator can engage the on switch region called enhancer and turn a gene on. But even if you manage to unravel the histones, if the enhancer region is blocked by certain molecules, for example, here we have this thing called a methyl group. It's one carbon attached to 3 hydrogen. If that thing is bound to your enhancer region, no matter what kind of stimulus you give your cells, it's not going to activate those genes because the activator is not going to recognize the switching region. And this is epigenetics. So you possess a perfectly functioning gene A, but you are unable to express it because due to some reason, the expression is blocked by some kind of modification on the DNA. And what does all this have to do with aging? Aging is the most complicated and still a very mysterious thing to humans. And everyone has to age. It's the fundamental rule of life, except for one kind of jellyfish. And here, this is a very creepy 5 minutes time-lapse animation that I recently discovered that shows a young girl transforming into an old lady in 5 minutes. And it's scary, creepy, and at the same time fascinating. And I recommend everyone to visit this site and watch it. And yeah, it's both a blessing and curse to age. And the biggest problem among humanity is for the first time in our history, we have more people aged above 60 years old and very less people being born. That means more people are going towards a very broken lifestyle. I'm calling broken lifestyle because aging or living at an elderly age doesn't necessarily mean that you're having a healthy lifestyle. A lot of them are going through a very severe disease phase once they're on the decline above 60 years old. So the goal of anti-aging is to allow people to live a moderately better life or healthier and more fit life after they reach the 60 years old threshold. And so we have this thing in medical reports. Usually they would say someone died of natural causes, which means sometimes it means they aged, so they died. But technically no one dies of natural causes. There's no such thing as, we're not computers to turn off. So if someone dies, it's because their organs failed. It's multiple organ failure or just a single organ. In this case, this is like the World Death by Cause from 2017. So people died because of cardiovascular diseases, cancer, respiratory diseases, dementia, digestive problems. You can see here, like most of them are some kind of diseases that happen later in life or caused by some uh, medical conditions like morbid obesity. And epigenetics is not what you inherit mostly, it's what you expose yourself to. There are a few factors from environment that affects your epigenome on how your body behaves and expresses certain genes. One of them is stress, both psychological and physical stress, nutrition, consumption of toxins. Here it's not technically poison, but things like alcohol, tobacco, uh, prescription medicine, and etc. And behavior is a tricky one because it's still a growing field within epigenetics. Epigenetics is a really young field, and behavioral epigenetics is even younger field. So this is kind of like out there now. And like I say, stress, lack of sleep, bad diet, and consumption of or overconsumption of things like alcohol will lead to dysregulation of your epigenome. For example, look at this person here. This person is an unidentified person from UK, and he's a truck driver for 25 years. He's 50 years old when the picture was taken, and you can clearly see that his right side is more aged than his left side. That's because for 25 years, when he was driving his truck, his right side of the face was always facing the sun, and the left side was always facing the shade. So the sun exposed side looked more aged than the protected side. And this is one of the perfect examples for epigenetics on how environment causes you to age faster. And he is in a high risk of a cancer formation too, because this damage will eventually lead to some kind of skin cancer. They did another study a few years ago where they exposed a young person's hand and an old person's hand to sunlight. After a while, they found that high methylation, like I explained before, of genes associated with cancer in the young person's hand. This means when a young person goes under the sun, cancer associated genes are not activated. But when an old person gets exposed to the same sun, they have low methylation, means they are expressing high levels of cancer associated genes. So as you age, you're more prone to getting cancer, not because of your genetics, but because of how your genes are being expressed. And as you age, you go through genomic instability, mitochondrial malfunction, metabolic imbalance, chronic inflammation, accumulation of dead cells, and stem cell exhaustion. So, common myth or misunderstanding is adult humans or humans in general do not have stem cells. We do have stem cells, like a very small amount, like a negligible amount. But as you age, even that is depleted. But with some kind of epigenetic intervention, we can go through a rejuvenation process to somewhat modify our genome to a younger state. So here you see a very compact version of a DNA with the histones. But as you age, this structure becomes very loose and you start expressing random genes. You start expressing random genes and you start inhibiting the expression of other random genes. So this leads to cancer, obesity, and pretty much degeneration of health. And so this is the most taunted ideology in the scientific world. Many people didn't accept it in the past, but now it's pretty proven. Caloric restriction or starvation is one good way of extending your lifespan. We have shown this in worms, flies, even mice, where if you restrict calorie or put them in starvation, you could actually extend their lifespan. But we didn't know if this is like a genetic cause or epigenetic cause, but now we have a pretty good uh, evidence that this is caused by epigenetics. Like here, if you think of aging in terms of clock, doing caloric restriction actually methylates or deactivates aging-related genes. And it actually acylates, meaning activates genes that are related to adult neurogenesis, regeneration of neurons, and antioxidant expression, which is very vital for anti-cancer properties. And both of these combined, they extend your lifespan, not only your lifespan, but also a healthier lifespan. So, could we cure aging? And if we could, should we cure aging? And what could possibly be the benefit? And so, like I said, it's not about living forever. That's impossible because living forever or immortality is not limited by biology. Immortality is limited by physics. Physically, no atoms can retain their shape forever. So immortality is out of the question. The idea here is to allow people to live a healthier lifespan at the end of their lifespan. One of the biggest problems with elderly people is they lose vision. And recently, this is actually a couple of months ago, scientists have shown that you could alter epigenetics to recover optic nerve regeneration so that old and aged mice can recover their vision. So you can see here on the top image, this is a damaged optic nerve. And at the bottom, the damaged optic nerve started to regrow. So the population that regrew the optic nerve to allow vision actually had their epigenetics reprogramming altered. As you can see here, the red spot here, this means a methylated region. When they're young, there are very few methylated region. When they get old, they have a lot of methylation. And the scientists expressed some other genes that modify this methylation that reverted the old mice to somewhat uh, express the unmethylated uh, part of the genome, just like the young mice. So what this did was, even without altering the DNA, automatically resulted in recovering the optic nerve that allowed the mice to see again. And this is promising since we don't have to directly genetically modify a human to make them better. We just have to alter the way the gene is expressed, and hopefully we can do it by some kind of drug in the future. That means you don't have to go through complicated uh, procedures. So I'm a huge fan of space colonization, and I think that's the direction that humans has to take. And there's a very good reason for that. So our population is increasing at an alarming rate, and there's no evidence that we're going to stop soon. Eventually, we will reach a plateau and start to decline, but that is a time when it's too late to save the species or the planet. A lot of argument I get with this is, why do we have to colonize a different planet and terraform it when we can just take care of our own planet Earth? So one of the reasons is phosphorus. Phosphorus is a chemical or a molecule that's very important for growth of plants. And without phosphorus, there are no vegetation on the planet. And the problem is we have depleted and are depleting the reserve phosphorus on our soil at an alarming rate. At this rate, we have about 100 years before we run out of phosphorus in the soil. After that, we're not going to get any vegetation. It doesn't matter if you're practicing veganism, it doesn't matter if you're practicing omnivorism or any kind of diet. If there's no vegetation, we're not going to be sustaining life on this planet. And the only way is to reduce number of humans. And you can't go all Thanos reducing half of the planet. So the other way is to deport humans willingly, of course, to go and colonize new worlds. And to do that, biggest problem is space radiation. space is filled with radiation and going there is actually a kind of a death sentence. But with epigenetics intervention, we could be able to sustain life even in space without landing on any rock or moon or planet. We could sustain life just in space stations. Or we could extend our lifespan long enough so that the spacefaring humans who migrate from Earth could, in their lifetime, reach a target planet or moon. This also allows for another topic called neoevolution. So humans, for the most part, are not following the natural selection law first introduced by Darwin, because we are not in sync with our nature at all. We're taking a different route towards our evolution. And my prediction is eventually we are reaching a singularity of half machine, half human. We are already there. Like, we are constantly relying on electronics, things that are not carbon based, things that are not biology based. And it's just a matter of time until we integrate this into our own system. So this is a buffer zone between a biological humans, Homo sapiens, and Homo whatever that comes after that. So yeah, we're in exciting times, and I'm really looking forward to the future. Unfortunately, I might not be there to see the future. It's frustrating, but we have to deal with it. But I would like to lay foundation to this topic of aging and lifespan extension so that humans can continue living in the future. I think I went pretty fast there. Thank you.
Thanks, Dave. Actually, you went 2 minutes over.
Oh, really?
Yeah.
Oh,
It was
wow.
good. I didn't want to stop you because of our doom and gloom of not enough phosphorus in the
I talked too much in the last slide because I thought I'm like slow. All right.
All right. So, no, but we still have plenty of time for Q&A. Go ahead and stop sharing your screen so we can get like everybody on here.
Oh, sure.
Thank you very much. It's exciting stuff. We will, let's see. Here we go. I'd like to open the floor for questions for everybody here. And Dave, your screen is still being shared, so.
Wait, you want me to unshare it?
Yeah, so we can get the grid out. In case there's people on the chat who want to ask questions,
right,
because we do have at least between me and Agath, we got a couple of bio people here as well. And then anybody on the YouTube chat, please type in your questions and Katie will read them out for you.
I think Matt has a question that he would like to ask. He'll let you get
good.
All right.
Matt.
Can you hear me?
Yep.
Cool. I was wondering if, like you mentioned that we can't, like immortality is basically unreachable because all atoms will change or decay over time. But say, for example, and tell me if I'm wrong, our body is constantly changing with atoms. Like I read somewhere that every 7 years, every cell in our body is a completely different process, for example. So if that were the case and we're replacing our bodies with cells all the time, if we keep those cells in good condition, technically, wouldn't we be able to recycle those cells over time? Sort of thing, therefore live forever?
Right, we would be able to, but I'm talking about, when I say that, it's like in a time of billions of years. Like when matter doesn't exist as matter anymore. So yeah, but you're right. If we could do that, because a jellyfish, I forgot the name of the jellyfish, does do that. After reaching adulthood, it goes back to its baby stage. I don't know what you call a baby jellyfish, but then it regenerates back to adulthood, and it became a pest because they're not dying and they're just growing like that and reproducing. So yeah, you're right. It is massively possible, but problem is we're just too complex.
Cool, thanks.
Yeah. By the way, the immortal jellyfish, the colloquial term is they're called immortal jellyfish.
Right.
Yeah, but there is a researcher actually at Kyodai who studies them.
Right. Oh wait, really?
Yeah, he's a really funny guy. He has like a song album out that sings about him. He wears a weird jellyfish hat. He's one of the
I should
quirky ones.
Right.
I had a question. I don't know if I can raise my hand or something.
Oh no, go ahead.
Hello. I've heard about telomeres that are supposed to be at the end of DNA, and I was wondering if you take that into account in your study. I think that's why clones cannot work, something like that.
Actually, our lab is also a telomere lab.
Hmm.
But yeah, you're right. For the longest time, we thought telomere.
Might want to explain what telomeres is for a second.
Right. So everyone saw chromosomes, right? So like I said, chromosomes are just like strings of DNA wound around this bead-like structure, but you need to keep them enclosed within the structure. So at the tip of the X, you have like an extra DNA that works like a cap. So the DNA is wound on itself, and then the last part of the DNA winds itself back like a cap so that everything is like intact within that X formation. But we know that our cells are always dividing. So when they divide, they also have to replicate the chromosomes so that the sister cells will have identical amount of chromosomes. So when you do that, the telomeres shorten slowly. Eventually they'll shorten so much that they cannot retain the structure, and the cell will just die because it cannot keep its control centers intact. So we thought, since telomeres are shortening when we age, it might be directly associated with aging. And back in 2005, they did a study where they extended mice telomere, and they found that it didn't do anything for aging, unfortunately. But here's another thing. Cancer cells are called immortal cells because as long as you give them nutrients, they'll just keep on living. They have incredibly long amount of telomeres, and the telomeres are always regenerating. So it plays some kind of role in cell immortality, but it doesn't directly correlate to aging. So yeah.
Okay, thank you.
Okay, I have a question from Rick from YouTube. He said, what percentage would you say is dictated by your DNA makeup when you are born versus influence on DNA through various stimulus as we age?
I see. That's really hard to tell because it really depends on stimulus. Maybe you have seen twins that are grown in different places will eventually, because when they're born, they're technically clones because they form a single egg, especially identical twins, or maybe just identical twins. But if you put them in separate places, their skin tone might change. One might be slightly obese than the other. Some might even look more aged or taller or shorter. So one fine example is height of a person. A person might be short, but not because of dwarfism, but because he's not getting enough nutrition. So it's really hard to pinpoint the, what do I call that? The percentage, because it really depends on what kind of environment you're living in. And that's the tricky part of epigenome because there's no way of quantifying it.
Yeah, that's so interesting. I didn't know that kind of environmental factors had anything to do with genetics.
Right, right. Yeah, so that's why it's a very young field. Like we're just slowly realizing that genes have very little to do.
Space and Earth as well.
So it changes your genome.
It doesn't.
Okay, if you
It doesn't. It just
I need to research.
So for example, skin color is the most amazing example of this. So we all have different skin color, and that's because of a mutation. Like our ancestors developed different mutation that allowed them to have different skin color and eye color. And usually people in the equator have darker skin tones like my ancestors compared to the ones at the northern hemisphere. But even if you're someone who had a lighter skin shade, if you go into a sun, you can get some kind of tan. So you'll be more like a golden color. But some people can't do that. But they do have the the gene that's required to produce melanin, but they don't produce melanin because recently we found out those gene areas can be methylated. So even having the gene doesn't matter because your activators cannot recognize the gene location. So yeah, your genome is straight. It's just that the expression is messed up. So that's why we all have the same genome. We're just like expressing it in so many various ways.
Okay, thank you. That's really interesting. I'm gonna do more research after.
Can I ask one thing?
Sure.
Yeah, it's really interesting story, and personally, I was curious about if lotions like sun skin and those, you know, like medical things, so also too, working or is not working.
Well, they work because like what do you call it? The sunscreen works as an artificial barrier.
Yeah.
It does work, but it doesn't do anything for your epigenetics, most likely.
Like Japanese people are really crazy about like putting sunscreen everywhere, right? Is it working really? I thought, yeah, because if it's like a genetically, you know, like claimed, then.
Right. So they need. So some people need the artificial lotion.
Yeah.
So the SPF can protect them from UV rays.
Okay, thank you.
Right.
All right. Well, yeah. Well, with that, I think that was a good way to call things off. Yes. Another round of applause. Another round of digital applause to Dave for a really interesting talk. If you have any more questions to him, we might type it in the chat or type it in the YouTube chat. We'll send him over to him. Maybe during the break time, he can keep answering. Is there if anybody has any glaring questions, is there a good place to reach you, Dave? Unless, you know, going to the going to your lab up at BioStudies.
Right. You can ask me on Twitter. So.
Cool.
Should I type your handle?
I will actually, if people can, I'll tag you on the Nerd Nite Kansai handle. So if you, for people who want to reach him on Twitter, then just go to our Twitter and he'll be on there. Excellent. All right. While I try to stall for time here, and I will keep talking so I can set up the 5-minute timer so that we can get started on our next one. All righty, so we will take a 5-minute break and then we will be back up here and talk about something very seasonal with Yuri.